morning and welcome to the first event in our Spotlighting Careers series looking at careers in science and technology. Today we're going to hear from Dr. Rossant, who is the President and Director of Scientific Research at the Gairdner Foundation. But first, we're going to have a message from Dr. Marek Radomski, the Vice Dean of Research for the College of Medicine at the University of Saskatchewan. The highly regarded and widely cited researcher, Dr. Marek Radomski, has had a commendable career in both academia and the pharmaceutical industry and has had a tremendous curiosity and passion for research. Dr. Radomski's inexhaustible enthusiasm for research and the lure of a challenge brought him to his current position as the Vice Dean of Research at the U of S College of Medicine. He took over as Vice Dean of Research in 2016, where he provides vision and leadership for all things research at the College of Medicine. Please welcome Dr. Marek Radomski.
Good morning and welcome. It is my honor and privilege to provide warm greetings to all of you on behalf of the University of Saskatchewan College of Medicine. Normally, the past years, we will welcome you in person in a wonderful organization, Vaccine and Disease Organization, Video Intervact. Unfortunately, this year, the COVID-19 pandemics have interrupted our plan, but I hope that next year we'll be able to resume a normal program. So the College of Medicine, University of Saskatchewan, a number of years ago, teamed up with the two great partners, the Gairdner Foundation, and you will hear all about the Gairdner Foundation, and its mission to popularize the science and bring the research to the attention of everyone. And Gairdner Foundation also rewards the great Canadian and not only international scholars with the very prestigious rewards. The second partner, the Saskatoon Industry Education Council, has been absolutely amazing in facilitating the school students coming and interacting with us at the University of Saskatchewan. Normally, this interaction will consist of the two parts. The first part is the lecture given by the Gairdner Foundation awardee, so the Gairdner Foundation CEO. And the second part, it's the amazing experience of going to the labs in Video Intervac and actually spending the half a day hands-on working with our scientists. This, as I said, it will not happen this year. However, I will truly hope that we will be able to do it next year. The Video Intervac is also an extraordinary organization being involved in the development of new vaccines. And in the times of the COVID-19, the development of new vaccine is such an important thing for the entire world and, of course, to us in Canada and Saskatoon. And the University of Saskatchewan Video Intervac scientists are at the forefront of this effort. So with this preamble, once again, I would like to welcome you to the University of Saskatchewan, virtually to our campus, and I hope that you will have a wonderful career day. Thank you.
Next, we will hear from Dr. Janet Rossant, Stem Cell Biologist and Scientific Leader. Dr. Rossant is a world leader in developmental and stem cell biology. Dr. Rossant has provided significant insight into how an embryo develops, how genes control development, and how pluripotent and other stem cells are established. Her research interests focus on understanding the genetic control of normal and abnormal development in the mouse embryo and its impact on human development and disease. By understanding the underpinning of early development in the mouse embryo, she has contributed to the understanding of the human embryo development and stem cell origins. Further, her research on genes controlling blood vessel development has defined novel pathways for new drug interventions in cancer. Throughout her career, Dr. Rossant has been a pioneer and innovator of new techniques to manipulate the mouse genome, establishing the mouse to become the preeminent model for understanding the function of the human genome sequence. So Dr. Rossant will give her presentation today, and then we will also have some time for question and answer from the audience. So if you have any questions for Dr. Rossant, please put them in the Q&A function, and we will try to address them at the end of her presentation. Please welcome Dr. Janet Rossant.
Hello, everyone, and it's great pleasure and honor to be here to talk to you today. In fact, I have visited Saskatoon before to give exactly this lecture with students at VIDO. So it isn't quite the same when I'm sitting in my back room doing this presentation, but I do hope that some of the science and some of the career advice that I have mixed together in my presentation will be of value to you, and I'm open to all sorts of questions and answers at the end. So I'm now going to share my screen. So give me a moment here. I've called my presentation The Winding Road to Becoming a Stem Cell Biologist. And what I'm going to do is take you through my career from the earliest stages and some of the steps along the way. And at each point, I'll point out how I keep coming back to some of the questions that you already heard that I'm interested in, which is how does an embryo develop? How do we all get here from a single cell, the zygote, to become the organism that we see ourselves today? And of course, in doing that, how can we derive stem cells from the different stages that can be useful in regenerative medicine? So it's a winding road. I started in the UK, as you can probably tell. I was born in Kent in the UK. I was the first person in my family to go to university. I studied biology at school, and nobody could give me advice from my family about what to do next, but I had a fantastic high school teacher, biology, who really encouraged me to go the whole way. And I hope that all of you who are thinking about science have found those amazing high school teachers who really can inspire you at a very important stage in your career. So with that in hand, I went to Oxford and I studied zoology. I then went on from Oxford to do a PhD in developmental biology at the University of Cambridge. So why did I become a developmental biologist? Well, it was actually during my undergraduate training at Oxford where I was taught by this guy here, John Gairdner. And John Gairdner is famous for cloning frogs. He was the person who showed that if you took a cell from an adult frog and put it back in the egg, as shown here, then you could actually get the DNA in the nucleus of that cell to reprogram development and make a frog. And so on the bottom, you see a number of little cloned frogs. And there's John in his frog room. I have to say, I was really excited by this work, the concept of how we can program development from the one cell through to development. But I didn't want to work on frogs, and you can perhaps tell why, because you'd have to work with these rather nasty, slimy creatures. So I moved from Oxford and started my PhD in Cambridge. But along the way, I should say that although I chose not to work on frogs, the work that John Gairdner did back when I was an undergraduate on demonstrating nuclear transfer in frogs won him the Nobel Prize in 2012, alongside a friend and collaborator of mine, Shinya Yamanaka from Japan, who was able to show that you actually didn't have to go back and do nuclear transfer, but you could reprogram adult cells in mammals to early development just by expressing a few specific genes. So these two very appropriately shared the Nobel Prize. So frogs were important, but I went on and worked with mice, and indeed Shinya worked on mice and then human cells. So not frogs, but mice. And I worked with Richard Gairdner, who's shown here, with his wife in their retirement in a beautiful place in the Yorkshire Dales with me. And Richard was the first person to really generate what we call mouse chimeras, where he could take cells using a complicated micromanipulation setup here and inject cells into my favorite stage, that picture you saw in the first image there, which is the blastocyst. And he showed that you could put cells into the blastocyst, and if they carried a genetic marker, make them black, you could end up with a mixed up mouse. that tool of mouse chimeras was extremely important, because it was that tool that enabled his lab, my lab, many other labs over the years to actually define what's going on in this so-called blastocyst. So here's a mouse blastocyst, size of a speck of dust, about 100 cells, about to implant in the uterus, so we can't get at it anymore. But at this point, it's really a beautiful structure, stained here with antibodies to specific genes, but it has 3 cell types. And what we were able to show then and in succeeding years since then, using the chimera approach, was that these 3 cell types gave rise to different structures later in development. The trophectoderm on the outside give rise to most of the placenta. This group of cells here, called the primitive endoderm, end up making what we call yolk sac endoderms, another sort of membrane that keeps the embryo happy in the uterus. And it's only a subset of little cells here that are actually the most important ones. These are the epiblast cells, and we call these pluripotent because they can give rise to the entire fetus. So these are the pluripotent cells of the embryo. They don't make the extra embryonic cell types, but they contain all the information to go from this little blob to a complete fetus and then a complete mouse. So that's what I was doing as a graduate student and postdoc in the UK. Then I moved and I came here. In 1977, I married a Canadian who I'd met at Cambridge, actually on the rowing team. It's another story. And we moved together back to Canada. I came at that point without a job, so career advice, don't do that. However, I was able to find a faculty position, a junior faculty position, or a 1-year position in Brock University, which is in St. Catharines. So at that point, it was very heavily undergraduate. It's really improved and extended its graduate research program since then. And I was a faculty member in the biology department. And I stayed there, continued my work until 1985. And during that period, I really was beginning to do what I continue to do and understand how we get to the blastocyst. So here's an egg, and here's the stages of development. The cell divides all the way through. They all look the same, and then gradually cells start to look a bit different by the 32 cell stage. Then we end up with a nice trophectoderm and a mixed up group of cells in the inner cell mass. Then finally, everything sorts itself out, and we get these cells that give rise to the structures I showed later. So at that point, I was doing a lot of experiments with the tools in hand to try to understand the cellular and molecular basis of these events. And here's just a couple of pictures. My lab group at that time and me with a mouse. I don't think you would see those kind of pictures of me with a mouse anymore, but there we are. We looked after our own mice. We had our own room. We had very limited resources, but we did some pretty cool and pretty important experiments even then. So how did I become a stem cell biologist? So that's how I'm a developmental biologist. I want to understand these structures in the mouse embryo. How did I become a stem cell biologist? Well, the blastocyst I just told you contains these pluripotent cells. And the question that I had in my mind all the way through these early stages and continuing on afterwards was whether we could actually make stem cells from the blastocyst. The blastocyst is so tiny, how are we going to understand it if all we have is these tiny little embryos? What I'd like to have are cell lines that grow and make lots and lots of cells so I could really study what's going on and use that as a model to understand the embryo itself. That was the idea. And jumping ahead many years, in fact, we can make stem cells from all those 3 cell types in the mouse blastocyst. And this has really been a powerful tool to understand early development and how cells talk to each other. The most famous stem cells from the blastocyst are embryonic stem cells. And I'm sure you've all heard about pluripotent embryonic stem cells in the human, but this is where they were first derived in the mouse. My lab did not do this. We were trying, we did not succeed. Other labs succeeded and set out the pathway for all of us to be able to derive embryonic stem cells. And they come from these epiblast cells. But my lab was able to show that we could derive trophoblast stem cells from the outer cells and Zen cells from the primitive endoderm. So now we have 3 cell types. They look quite different. They proliferate indefinitely. And interestingly, when you go back to that blastocyst chimera that I showed you Richard developed in the early stages, you put those cells back in the blastocyst carrying fluorescent or blue markers, then you see that ES cells make fetal cells, but not yolk sac and placenta. TS cells contribute to the placenta, and Zen cells make this other membrane, the yolk sac. So they're really remembering where they came from. So that's been a great tool. So that was going on while I was still at Brock. But then after 8 very nice years at Brock University, I wanted to test myself because I had tenure at Brock. I was doing a lot of teaching, and I wanted to move into a research-intensive environment. And at that point, the Langenfeld Research Institute at Mount Sinai in Toronto just opened, and I was one of the founding faculty. So I joined the Langenfeld in 1985, great set of colleagues, and stayed there for 20 years. And during that period, we did a whole load of exciting things with mouse embryonic stem cells. One of the most exciting, my favorite experiment I've ever done is this one, where we asked if ES cells are actually pluripotent, could we make a mouse that's entirely derived from stem cells that we grew in a petri dish? So this was work done with my colleague at the Langenfeld, Andras Nash, and here we are, we're up at the top of a very tall tower in Hong Kong, if anybody's ever been there. And he looks a bit nervous about being up so high. I'm obviously quite relaxed. Anyway, we had a wonderful time working together. In fact, we still collaborate on a lot of experiments. But what we did was this experiment here. I'm not going to go through the details. Suffice it to say that what we realized is that we could do a complementation system, another kind of chimera. Because we knew that ES cells, I told you, they make the embryo, but they don't make placenta and yolk sac. And we knew that tetraploid embryos, which have twice the normal amount of DNA, which we can do by fusing cells together, it had been known in the literature that they make yolk sac and placenta, but they don't like making the fetus. So if you stick them together, as shown here, then indeed what you see is that, of course, you're basically able to make a placenta from the green cells, and the entire fetus is derived from the ES cells. So that's good. Can we actually make a mouse? And the answer is yes, we can. And here they are in the same experiment. This is a whole litter of little mice. They're glowing green because they're expressing GFP, and they're entirely derived from stem cells that we grew in culture. And they're perfectly viable. They grow up, they breed, and they're perfectly happy. So ES cells can make mice. That attests to the fact that these cells can be grown in culture for a long time and retain capacity. So if the mouse cells can make a whole mouse, that really invigorated the search for generating human embryonic stem cells. Not obviously to do the experiment I just showed you, but because they would be totally pluripotent, they could be an enormously important source of cells for regenerative medicine, for repairing tissues from degenerative diseases or injury. And so, again, not our lab, but Jamie Thompson in 1998 was the first person to derive human embryonic stem cells, essentially the same way as in mouse, from embryos that were left over from IVF programs that would otherwise be discarded. Was able to get, again, proliferative, ongoing cells that had very extensive pluripotent capacity. So this is just an example of some of the different tissues that you can get from differentiating human embryonic stem cells in culture. And then, of course, the idea is that you grow them into tissues that could then be used to repair human disease. So we've been around, obviously, since 1998, a lot of hype early on about pluripotent embryonic stem cells and their ability to really move to therapy. So where are we now? Isn't it time that we started to see these pluripotent cells coming to the clinic? And I would say the answer is we are getting there. It really is moving. The last few years, we are seeing that human pluripotent stem cell therapies are moving forward to the clinic. These are just my top list of those where there's really already clinical trials undergoing, trials about to begin. So for spinal cord injury, not replacing the nerves, but replacing the cells that wrap the damaged axons. So a couple of trials underway with this approach. Macular degeneration, where the retinal pigment cells die out, turns out to be very easy to make those cells from ES cells, and the trials underway in Europe and in North America look quite promising. Parkinson's, we know that you can get some alleviation of symptoms if you transplant dopamine producing cells, and there's a couple of international consortia actually close to moving to clinical trials with this. I think this is really going to be extremely important. Type 1 diabetes. We know that if you transplant islets, that contain the cells that make insulin into patients with diabetes. You can get them off insulin, but you can't get enough islets, enough donors. There are trials underway now with insulin-producing cells from human ES cells. And then close to home, it's a very exciting preclinical data, not yet in trials, in replacing damaged heart cells after heart disease of various sorts. There's a group at the University Health Network here in Toronto who are really pushing this extremely hard in association with a couple of companies. So it's moving forward. It's really, really exciting. So to be able to think about how looking at the embryo, identifying pluripotent cells, taking those and making stem cells, and now this is moving to the clinic. So that was all going on during the years that I was at Langenfeld. And then I crossed the road and took on a more administrative job while still maintaining my lab. And I became chief of research at the Hospital for Sick Children in Toronto. Now that's a research institute that has about 250 investigators, 2,000 people working there on everything from molecular structure and function right through to population health and health outcomes. So I learned a lot because I had to try to direct and advise a whole group of investigators and really help their careers. So that was a very rewarding time as well. But when I was acting as chief of research, I didn't get into big stem cell clinical trials or anything like that. It was going to be too difficult. I actually went back to the embryo and realized that there were still questions that I wanted to ask about how you make a blastocyst. And those were going to be able to be answered, again, because of new tools. We have new tools that come up every 10 years or so to help us go back and ask old questions in new ways. So what we really wanted to do was, instead of just having static images of what the embryo is doing, it's a dynamic process. That cell is, those embryos are dividing, reorganizing, making structures, making cavities. What happens to the genes that we think are important for development during that process? So to do that, we need two things: improved single cell analysis of gene expression, and that's come about because of a great deal of investment and excitement in single cell RNA-seq, so we can really assess each cell and what it's expressing in terms of genes. But then more importantly, from what we want to do, which is look at the embryos, we can follow cell fate by tagging those genes and with fluorescent tags and watching how the embryo develops in vitro through live imaging. And how did we do all that? Well, I said there were new tools, and the biggest new tool on the block is gene editing, so CRISPR-Cas9 gene editing. And I don't know whether you've gone into the details of this, but again, you've probably seen it because the Nobel Prize in Chemistry just a couple of weeks ago now went to the people who developed this tool. And it's basically an enzyme system. So there's an enzyme called Cas9, which binds to a guide RNA that identifies specific sequences in the DNA. And when these are brought together with a few other bits and pieces, you get a very specific cut. It tells you where to cut the DNA at a precise moment of position. You can cut it, make a mutation, and once you've cut it, you can also introduce other donor DNA into the site and make alterations. It is from bacteria. There have been other systems before, but this particular system is very specific, very efficient, and essentially cheap. So it's widely used now for genetically modified plants, animals, bacteria, fungi, and of course, we use it a lot in the lab to make genetically modified mice. And we do that with a technique that two of my postdocs developed, where we take all the bits and pieces that are required for gene editing and inject them into the two-cell embryo. Both cells of the two-cell embryo turns out to be then extremely efficient to get these, this construct to cut and mutate and introduce fluorescent tags into our gene of interest, and then make the mice, breed them. Now we've got a mouse line that we can use to study development. So I'll just show you one image where we tagged two of the genes. One is involved in making trophectodome, and one of them is involved in making those inside cells in the blastocyst. You can watch them actually come on. So the green is CDX2, it's marking the outer cells that are going to make the placenta. And then it becomes very strong in the outside, turns down in the inside cells. And as it goes down, we start to see red coming up because the inside cells are now expressing SOX2, which is required to specify the inner cell mass. And you can see that it's there's no overlap there. It's a really segregated process dependent on polarity and position. So we're learning a lot from this kind of approach to understanding the embryo, back to understanding the mouse embryo. And there's still lots to be learned. So I spent 10 years as chief of research and it was very rewarding, but 10 years is enough. And so in 2015, I stepped down from my role as chief of research, maintained my lab at SickKids. And then in 2016, I had the opportunity to become president of the Gairdner Foundation. And that's why I'm here today, although I'm also here because I did win a Gairdner Award early on. I was so pleased with getting the award, I just took over the foundation. And this is actually a great job too, because the Gairdner Awards are internationally recognized awards for the very best biomedical research that's going on worldwide that has impact on human health. And the Gairdner Awards, since 1957, when they first came about, have been over 300 given out. And as a measure of what we're recognizing with the Gairdner Award, 95 of those have gone on to receive the Nobel Prize. And of course, what we do at Gairdner is not just give prizes. We are here to engage students and public, policymakers, and everyone about the importance of research and the excitement of research. So that's the part of the Gairdner mission that goes beyond just the awards. And of course, we bring all these laureates to Canada, usually in person, this year virtually. They're all actually across Canada right now giving lectures. And we bring them together. And it's an opportunity for Canadian scientists and students and trainees and high school students to hear great science and hear great people. So just to point out that, yes, indeed, we do recognize people who get Nobel Prizes. So this was my first Gairdner Gala in 2016, when we did recognize CRISPR-Cas9, so 4 years ahead of the Nobel Prize. And it's what I call the gene editing quintet. These 5 people in the middle were recognized for their contributions. Rodolphe Barango and Philip Horvath for the bacterial work, and then Jennifer Doudna and Emmanuel Charpentier for turning it into a biochemical tool that would work in other cell types, and Feng Zhang, who really democratized it and made it into something that we could all use very easily and help the world become CRISPR-Cas9 experts. So that was great fun. And of course, as I said already, Doudna and Charpentier won the Nobel Prize in chemistry for this work just very recently. So it's a great reward. So, but you know, when you talk about CRISPR-Cas9 gene editing, well, there's other things that come up. It's great to make genetically modified agricultural products. People are working to make, to try and make trees, for example, that would carbon capture better, plants that would be resistant to drought as we go into global warming. There's lots of fantastic applications for CRISPR. And it's going to be very important for treating genetic diseases in humans, because the efficiency means that it should be possible to actually correct genes in tissues in patients who have specific genetic diseases. So for example, sickle cell anemia, which is a blood disease, there are trials underway with CRISPR approaches to try to reactivate the globin gene expression in those patients. There is an opportunity to treat a disease like cystic fibrosis, where the lung cells are blocked because they produce excess mucus. We know what the gene is, and there's certainly ways you could think about fixing the gene in the airways. So lots of excitement. But that's what we call somatic gene therapy. That's taking patients who have a genetic disease and trying to genetically repair and enhance their survival. What has become more in your face, as it were, what has become in the public domain is this concept, well, if this is so efficient, maybe instead of treating people who have diseases, why don't we just prevent it by modifying the genome in the embryo? Back in those stages, I showed you how we do it in the mouse. We inject into a 2-cell embryo. We could do that in the human embryo and then transfer it back. And now you would have a human embryo that lacked the specific bad gene. So this image comes off the internet, and I modified it a little bit. These are the sort of things that people talk about with germline gene editing. You know, could we improve IQ? Well, yes, but we don't really know the genetic basis. You could potentially affect gene predisposition to disease. I think I know how to make a better sprinter. We know some of the genes that control muscle development, so could be done. But really, if you're going to do this at all, this is what we would call gene enhancement. And every working group that's looked at this has said, we are not ready. We do not want to go down this path. This is not appropriate use of gene editing technology. But could it be used to prevent cystic fibrosis or Huntington's, serious single gene defects? So that's been the discussion. And there've been a number of working groups looking at this. The first sort of public discussion was when scientists got together with lawyers, with patient advocates, with policymakers, and discussed the future of gene editing, particularly in the germline. This was the discussion in 2015. Here's me sitting on the stage with a couple of other very well-known gene editing experts. And after that discussion, after that summit, there was a statement put out, which you can read here, which basically says it would be irresponsible, it's a very strong word, it would be irresponsible to proceed with germline editing until we know whether it's safe, whether it's really efficient, and really assessing whether there are other ways of getting the same result. And of course, that this has to be something that society as a whole thinks is an appropriate move forward, and there should be appropriate regulatory oversight. At present, 2015, these criteria have not been met. Well, more discussion groups. I was on a working group of the national academies in the US that came out with a longer report that basically came out with much the same conclusions. And then this happened. So in 2018, a Chinese scientist, Yang He, claimed to have produced twins by gene editing in the embryo, the gene CCR5, that if mutated, the babies would be resistant to HIV infection. Well, first of all, that is an enhancement. It's not a necessary intervention. There are many other ways to prevent HIV infection. And certainly this is not the first use of germline editing that anyone would recommend. It caused a big fuss in China and around the world. And in fact, he was fired from the university and ended up doing a small prison sentence because he had actually broken several different guidelines and regulations in China. But it sort of got the world going again. And there are two working groups still looking at this issue. And one of them just came out. This was a working group from the National Academy in the US and the Royal Society in London. I was on the oversight committee of this, so got to look at the overall report as it was being put together at the end. And essentially, here we are, 2020. We know a lot more about how CRISPR works and how it doesn't work, but we still don't think that this is safe enough to really even think about germline editing human embryos. It hasn't been proven. And there really are very few cases where you would think this is something that you would want to take through to the clinic. So I would say that despite this being a very popular discussion, this is not the most important use of CRISPR, but it is one where, as a scientist, I think it's very important for me and other scientists to get involved, provide some background on the science and provide some insight to help society, lawmakers, patient advocates, and other groups come up with their consensus about when and if this should ever go forward. So that's sort of how I got to be a stem cell biologist from a developmental biologist, and how you get involved in ethical debates that you really didn't expect to get involved with. So here I am. I'm at the Gairdner Foundation. My lab is getting smaller, but I'm still pretty excited. So what gets me up in the morning? I just think this is the most exciting time ever to be in biomedical research. The tools and technologies we have to address fundamental questions of development, of cancer, of organ formation, we have incredible tools. And as a scientist, the thrill of discovery, that moment when you look down the microscope and you see something that nobody has ever seen before, never goes away. And it's also very exciting because as a fundamental researcher, it's very exciting to me and to others to see how our research is having an impact in the clinic, in cancer, in genomics, in regenerative medicine. And it is a great opportunity. I really enjoy doing these kind of lectures, and through the Gairdner Foundation, I have more and more opportunities to try to pass on the excitement of science to the public and to students. And I do feel a responsibility to get involved in the ethical issues and help define the boundaries. So what about you? So that's my career and why I'm interested in science. I just love discovering how an embryo works, but it can take you in different directions. Why should you think about careers in science going forward? Well, I'm not necessarily saying that you should become an academic researcher like me. That's an exciting career if you find something that you have passion and you can work out how to really explore it in exciting ways. It's a lot of fun, but it's not for everyone. It's hard work. There's a lot of times when experiments just don't work. Mostly they don't work. So, you know, there sounds like it's very easy, everything just comes easy, and you find this and that. There's many years even when nothing seems to work. But even if you're not thinking about a research career, everybody needs to be science literate these days, because it's critical in this age of false news and internet and conspiracy theories that science, critical thinking, can be applied to the world around us. So whatever you're doing, science is going to be an important part of your future career. You need to find and follow your own passion. But science training is not necessarily going to make you an academic researcher, but science training is a great training for many different directions. And over the years, I've had about 80 or so trainees come through my lab, and many of them are research scientists, but they're also science writers. They are editors of journals, they're teachers, they're entrepreneurs. One of my postdocs has a quite successful biotech company, lawyers, doctors, patent agents. Many ways you can use science, but science is a great way to develop your training and a great way to find careers. So I'm just going to end with a couple of thoughts, which I'm happy to answer questions on, about women in science and diversity in science. I am a woman scientist. And as the field that I'm in, developmental biology and stem cell science, I've been very lucky, I would say, because these are fields where there has been consistently a high percentage of successful females, which means that there are good role models. That makes it welcoming and supportive field. I don't go into a room and find myself the only woman in the room, but there are fields still in science where you can find that. And of course, not just as a woman, if we extend this and look at diversity and look, for example, at people of color or indigenous people, there's still very underrepresented in all aspects of science. And it's not really necessarily getting any better, which worries me. This is just one example. There's many different kinds of data you can pull together on this. But it's been estimated that in 2018, and it's no different in 2020, that future jobs, you're going to need some science training, STEM, science, technology, engineering, mathematics. In the engineering and computer fields, the proportion of women is extraordinarily low. It's actually lower than it was in the 1960s. And if we look generally at the STEM workforce, we see that overall the workforce in Canada, about 50%, women are 50% of the workforce, but they're only about 25% of the science-related workforce. And there's a big dropout during the first 10 years. This means that we are not making the field open and welcoming to those people that we need, because we need diverse voices. We've got big global problems coming up, and if they're not only going to be solved by white men, we need women and we need diversity at the table, because it brings diverse skills and opinions, and that's going to be necessary for our future. So we've got to do better, and we all, and that's particularly those of us who are in science, need to break down these barriers, because we need to be sure that you, the leaders of the future, can succeed. So that's it. I've had a great journey. It's not finished, because there's so many challenges still ahead and so many opportunities to work in science and engage the next generation. Thank you.
All right. Thank you, Dr. Rossant. That was a great presentation that covered many of the questions that I see already in the thread, but we're going to go through some question and answer right now with the students. So the first question that we had asked is, what kind of student makes a great scientist?
Oh, that's a good question. That's a tough one. So I sort of said something about that, I guess, because I think that it's having curiosity. That's really probably the most important thing. If you're going to be a scientist, a research scientist, you have to have curiosity. You have to wonder how life works, or how the universe works, depending on what level of science you're going to be looking at. Or how does a computer work? You've got to be curious. So I think curiosity, number one, not accepting the status quo, challenging the status quo, because science is about testing hypotheses, about saying, is this really right? So a hypothesis is something you try to disprove. It's something people don't always understand about science. We're not trying to prove something, we're trying to disprove it, because only that way will we really understand how it works. So I think curiosity, not accepting the status quo, and dedication. I mean, it's hard. If you're going into research, it's a hard road, and it takes a lot of time and a lot of effort to go down that pathway. So commitment and dedication is important too.
Right. Our next question is, what high school classes should I be taking or focusing on if I want to get into science, specifically research?
So at high school, I think you have the opportunity. It's too soon. You don't know. First of all, you don't want to do research. Secondly, you don't even know what area of research you want to do. So it's important to take a broad selection of science courses. It's always good to have some math in the mix. So just keeping a broad selection of science courses at hand. And even at university, you don't really need to specialize very early on. You can, again, try out different areas. When I was a high school student, and that was a long time ago, and it was in the UK, where in the UK you have this, even at high school, you get this very much channeled very early on into a very specific pathway. So, when I was 16, you had to choose for the last 2 years of school to take 3 courses, only 3. And so if you don't, you have to decide, am I going to be a scientist or a historian or a English literature major? You have to decide at that point. And so I chose science, but I wasn't sure if I wanted to do chemistry or biology. So I did both, took courses in both, and I did math, which I was bad at. But you do need, but having said that, you should take some math courses. So I think but I think that's a bad way, because today science itself is so interdisciplinary. So in my field, for example, I came in at it through the biology angle. Other people, particularly now, are coming at it through engineering. Bioengineering is really important, to be able to take those stem cells and make them into the right cell types in bioreactors and all of that, the rest of that. There are physicists and computer scientists who are helping explore the genome and how all those different changes in the genome are played out in developing cancer, for example. So you never know. You need to come in from different angles. So that's not very helpful, is it? Just keep going. Do science. I think you're somebody. You also asked about doing stuff that outside of school that's going to help you in research. So yeah, I mean, I think that there are opportunities for high school students to do sort of small interns in labs at universities or places like Vido. Explore those. It's a great opportunity to play around with research, and particularly when you get to be an undergraduate, there are many summer student programs at all the universities across the country where you can spend the summer doing research in a lab, and that's a really great opportunity to see whether research is for you. Can you put up with all the disappointments for that one experiment that might work and probably won't work when you're only there in the summer? So I think that's another very important thing to do.
Awesome. Let's go to our question. What is your favorite part of being a stem cell biologist?
I think it's really Two things. One, the people in the field, and this is another great thing about being a scientist. So I said it's very rewarding, you know, you've got your passion, you're finding out things, and every day there's something new to see, and at least every month there's something new to see. But you know, science is a social undertaking. So the best thing I find about being a stem cell biologist is all the other stem cell biologists that I can meet here and around the world. In this COVID time, those conferences and those visits where we can all get together and exchange ideas have changed a lot. We're having Zoom conferences. They work to a degree. And you know, this is great. We're talking to each other like this, but it'd be so much better if we were there all together. And that's one of the things that is definitely lost during this pandemic, but it will be back. But yes, the science and the social interactions, which is how you drive new collaborations and new ways of thinking about science. So that's I think very important. So that was one. Oh, the other thing is that stem cell science, A, has great biology, and B, truly does have potential to cure and treat human disease. So it's very satisfying to think that what we do, not directly, but indirectly, can help humans.
Awesome. What is the best advice you've received that stuck with you throughout your career?
Okay, so that's a good one. I think the advice that I have received is the one that I just gave to you, and it's fairly common, but it's true. You really have to find your passion. And whether your passion is research, whether your passion is science at all, maybe your passion is something else. But if you're going to have a career that's going to be satisfying for you, you're going to have to spend a bit of time playing with different pathways and then finding out what it is that really gets you up in the morning. So what gets you up in the morning is probably the best measure of the career that you should go with. Some people say, you know, what you're thinking about in the shower, that's another one too. Thinking about mouse embryos in the shower, you're probably a mouse developmental biologist.
Okay, and that takes us to our next final question. Why did you choose to work with mice?
I gave you a little clue to that at the beginning of my talk when I said that I became a developmental biologist because I was taught by John Gairdner, who's a fantastic scientist and a great friend, I have to say. But I didn't like frogs. Well, horrible. You go into that room because, you know, we were working in the lab. We'd go into the room and do some stuff with the frogs. They were slimy, they smelled, and you feed them. At that point, they were feeding chopped liver. I'm presuming they have some better, more pellets or something now, but it was just disgusting. But also, I'm full of undergraduate arrogance that I thought this frog stuff was fine, but it would all pass by and it'd be over in a couple of years. And the real question was the mouse. And I got one lecture from a mouse developmental biologist in that course. And that's what turned me onto the mouse. And why the mouse? A, it was unexplored. So there's opportunities to do something really new. And B, the one I just said, that potential applications to humans were that much closer than if you were working on frogs. I was definitely wrong about frogs. I mean, John did win the Nobel Prize after all. I have not won the Nobel Prize. So I was wrong there. But that's how it became, why I felt the mouse was a really great system. And I've stuck with it, although we have worked with human embryonic stem cells and other cells types as well.
We have some more questions coming in. One says, you've mentioned how things aren't always perfect in research. What role do mistakes and failure play in scientific research?
Well, learning from your mistakes and failure is absolutely key for scientific research and probably for a number of other careers as well. But you have to learn from them. If all you get is a failure, my experiment didn't work, I'm just going to go home and have a beer, that's not getting forward. And you have to look at what you did and you have to say, well, that was a failure. How could I make it better? And I think it's that part that is really, really important. So that's the learning from failures and then learning how to move forward from those failures is really, really important.
Awesome. And do you need to have a graduate degree to become a science researcher?
Yes, you do. It depends, again, what level of researcher you're looking at. If you want to be a university researcher like me, yes, you would do a PhD, and you would probably also do what we call a postdoctoral fellowship. So you do an additional sort of semi-independent training where you join a bigger lab for a couple of years to really define what your passion is, what your area of research is that you would then hope to take to get an appointment in another university around the world. So yes, you do. However, there are ways you can work in research. You always need an undergraduate degree. There's something that we call a master's program. It's a shorter graduate training that can be really quite useful in getting you into working in a research group or working in industry and other environments. You can also become a technician, and there are college training programs that will train you to work specifically, for example, with stem cell cultures. So if you're taking stem cells to the clinic, there's actually a crying need right now for people who can actually grow these cells and carefully maintain them in culture to a point where they could actually be used in the clinic. So there's different levels. You certainly need an undergraduate science degree and some form of postgraduate training.
Okay. Awesome. Well, I think that takes us to the end of our questions. Thank you so much for joining us this morning. It was great to hear about your research, and it was great to hear about your career path. I know you had some very inspirational words, and in our office here as we're watching, we saw a lot of thumbs up from some things you were saying, and it really connects with what we do at the SIEC with letting students explore different career paths and ultimately finding their passion that they can turn into a career. So thank you so much for joining us. I am going to share my screen because we have one little activity. What am I? Okay. Just wanted to make sure I was sharing the right screen. So at the SIEC, we typically host all of our events in face-to-face formats. However, this year we are going to a virtual format, but we still want to ensure that we're meeting the needs of the students that we work with. So I have a survey. So if students are watching this presentation right now, we ask that you take out your phones. Sorry, teachers, if you don't like them to have their phones out in class, but we would love some feedback on this event. So we have a QR code, and just for a little bit of enticement to give us some feedback, we love to give out prizes. So if you provide us with some feedback, we will be doing a draw to give away some gift cards. So please let us know what you thought of the presentation today. So I'll let that sit up for a couple seconds while you guys can scan it. And if you enjoyed hearing from Dr. Rossant today, or if you maybe thought, scientific research might not be for me, but I do still have interest in science and technology, we have a few more events happening this week. Tomorrow night, we have an evening event where we are going to look at careers in agriculture and bioresources. So we will have a representative from the U of S College of Agriculture, and we'll have a representative from Sask Polytech Bioscience, and they're going to share some of the post-secondary options they have. As well, we're going to hear from a post-secondary student and an industry professional who's going to share their career journey. And on Thursday, we have 2 live events taking place. Everyone has probably heard the word COVID-19 too many times to count in the past 7 or 8 months, but we are going to talk with Dr. Volker Gertz, who is the president and CEO of Vito Intervac. And Vito Intervac is a local organization here in Saskatoon at the University of Saskatchewan, and they are leading the research in COVID-19 vaccine. And so we are going to hear from him, and he wants to take your questions, and he wants to answer any questions you may have about COVID-19, about the vaccine, or about creating vaccines in general. So we highly recommend that you watch one of those or tune into one of those live events. And then teachers, we also have some resources for you on our website. We have teamed up with Vito Intervac and Egg in the Classroom, who have provided some lab activities that you can do with your students. And we also have some lab activities from the CDC that deal with pandemics and outbreaks. And so they can be very relevant to what we're dealing with in our society today. So check out our website to find out some more information about the different programs and the different resources and activities we have going on there. And finally, if you would like to keep up to date with us, we ask that you follow us on social media. We post all of our events that we're hosting throughout the year on our social media platforms. So follow us, stay up to date. This month we're looking at science and technology, and maybe after looking at these presentations you realize, that's not the career for me. Well, that's okay, because every month we're going to focus on a different sector. And so next month we're going to look at mining and manufacturing. Maybe that's something you're more interested in. So please give us a follow, stay up to date, and hopefully we'll see you at our events again soon. Thank you everyone for tuning in today, and we look forward to seeing you at our future events.